So mRNA processing consists of 3 steps, and it only happens in eukaryotes. Prokaryotes don't process their mRNAs. But in eukaryotes, you'll have the addition of a cap. The cap is a special attachment of what's called a 7-methylguanine. All right. And 7-methylguanine nucleotide will be attached in sort of a backwards fashion at the 5' end of the RNA chain. The other thing that happens during processing is called the addition of the poly-A tail. There's a certain sequence close to the end of the mRNA. In this picture it's sort of in the middle, but imagine it close to the end on this side, that is AAU, AAA. And there's an enzyme that looks for that sequence and then trims the mRNA a little bit past that site. And to the trimmed end, the enzyme adds a long stretch of adenine nucleotides. So, maybe 100 or more adenine nucleotides. That's called polyadenylation. So polyadenylation creates what's known as the poly-A tail. mRNAs from eukaryotes have this long poly-A tail at the 3' end of the molecule. And the third thing that happens, first the addition of the cap, then the addition of the poly-A tail. The last thing that happens for processing of the pre-mRNAs is splicing out the introns. And when the introns are spliced out, what's interesting is during the process of splicing, the introns become connected to themselves in what's called the lariat shape, which is like a lasso, like a cowboy uses. And so under the microscope you can actually see in the nucleus, you can see, if you have a powerful microscope, you can actually see these little lariats appearing in the process of splicing. And so these intron segments will be broken down into their individual nucleotide components, and the nucleotides can be recycled, reused. But in the end you get splicing together of all the exons into one continuous coding region. Most eukaryotic genes have some introns, although the number of introns can vary quite a bit. You can have anywhere, actually some genes have none. Most of them have at least some introns, and at the very maximum, I think 204 is the most number of introns that they've found in any one gene. 200 and something. But anyway, so that's pretty extreme. I would say typical would be in the 10 to 15 region would be pretty typical. So kind of a handful of introns is kind of typical. But there's lots of variation in genes. One thing that happens that I don't know, maybe 15 years ago we thought was pretty rare is something called alternative splicing. It turns out that it's not rare. It's something that a lot of genes do. If you have several exons in a gene, this is just 3, but if you have several exons, it's possible and actually quite common for the exons to be spliced together in different combinations, and they give you slightly different gene product or mRNA products, and slightly different proteins will be translated from those. So in this mRNA we got exon 1 spliced to exon 2 spliced to exon 3, but in this mRNA we just have exon 1 directly spliced to exon 3, and the middle exon 2 has been excluded. And so you can get these alternative splice variants among the mRNAs of the same gene. And so those are called splice variants. And then when these are translated, you end up with different, what we call isoforms of the same protein. So an isoform are molecules of virtually the same protein except translated from different splice variants. So that's kind of interesting, because then what you end up with is one gene that can be used in different ways to create somewhat different protein products. So I will let you look at the overview of eukaryotic gene expression after you watch this lecture segment. tRNAs are also copied from genes. tRNA genes are actually one of your types of a multi-copy gene that's considered repetitive. So tRNAs are encoded by tRNA genes and they're produced by transcription. They don't have any codons. They will actually fold into a secondary structure that looks like this cruciform, and they have to be trimmed down, and they actually contain some weird bases, including ribothiamine. So it's an RNA version that has thymine. Something called pseudouridine, which is sometimes shown with the Greek letter psi, and something called inosine, which is usually indicated with the letter I. And so there's some weird nitrogenous bases in tRNAs, and that creates some interesting things that we'll look at in Chapter 11 in terms of how the base pairing happens between a tRNA and an mRNA. But in three-dimensional form, you see a shape that looks like something on the left of this picture. This is a space-filling model of tRNA. This is sort of a ribbon model. And then on the right side, you see sort of a flat two-dimensional image of what the tRNA looks like if you flattened it out. But the book will use a shape that looks like this in the lower left-hand corner here. That's going to represent tRNA in the figures in the next chapter. So what is a gene? Well, most people would say a gene is an entire transcription unit. That would include the promoter sequences, which aren't copied into the RNA, the terminator sequences, some of which aren't copied into the RNA, the introns, which are trimmed off, and anything else that's trimmed off. So a gene is in the chromosome. A gene is the whole unit that is needed to get transcription to happen. Not all RNAs are mRNAs, so not all gene products are proteins. A lot of the RNA types that we looked at in that list are what are called untranslated RNAs. They do their own thing in the cell, and that's really shifted our paradigm, because we used to think that, DNA is copied into RNA and all RNAs are needed to make proteins. Well, it turns out that a reasonable sized group of RNAs doesn't ever have anything to do with the production of protein. So that's really changed kind of the way we think about the central dogma of biology. Okay, and the last section in this chapter is about RNA interference, another type of RNA. It's called short interfering RNA and microRNA. So siRNA stands for short interfering, and miRNA stands for microRNA. They're both very small RNAs. They're copied from a gene, or they can come from somewhere else, like a transposon or a virus. And really what they do is they stick to mRNAs that are being made in the cell. So they're complementary to mRNAs that are being made in the cell. And when they complementary base pair to an mRNA, then that marks that duplex for destruction. So if a short interfering RNA or a microRNA sticks to any kind of mRNA, then that mRNA won't be used to make a protein. It'll block it. Okay, so in some cases it just blocks it from being translated, and in some cases it marks it for destruction. But in the end, the effect is the same. You won't get any protein product from that mRNA. So short interfering RNAs and microRNAs basically serve to block expression of mRNAs. The model organism in this chapter, which is described as the nematode worm, C. elegans. C. elegans is a very small organism, but it has a very short generation time, and it's very easy to have a lot of them in a small amount of space in the laboratory. They have a very short life cycle, as you can see here. The contributions to genetics have mostly had to do with development and apoptosis, which is involved with development, programmed cell death. Because all of the cells in C. elegans are programmed from the very beginning. The cells are programmed to become certain parts of the body. So there's a very hardwired genetic development of every cell in the worm body, in the nematode worm body. And that's called a determinate type of development. Now, humans, we don't have determinate development, we have indeterminate development. But a worm has a very determinate development, meaning every cell from the time it forms knows exactly to what structure it will eventually be part of as it divides. There's no adjustment that can be made. So what they did was they took worms at every stage of development and they used lasers to kill individual cells one at a time, and then they would see what happened as that worm developed, what structure is missing. So it's called laser ablation. And by doing that, they were able to map out the fate of every cell in the developing embryo, which was really kind of fabulous. However, it doesn't really translate to human development directly, because humans, if a cell dies during human development, then the cell next to it will divide to replace it, and it keeps going. So you won't be missing any structures in the end. But anyway, the worm was used to figure out a specific pathway of development, which is called determinate development. And that's the end of our Chapter 10 lecture.